Okay, welcome everybody to the second in the series this year, microTAStic. You can see on the slide we're sharing right now just a little forecast of what's to come. So if if you haven't put us on your list of seminars to pay attention to, you can scan the QR code and get more information. We have, in addition to the speakers we have today, we have some awesome speakers coming up for the next sessions in the series as well. So just to jump right in, so today we have two speakers, and our first speaker is one of the selected speakers from the journal series. And today we'll be hearing from Dr. Stephen Vasilescu. He is a biomedical engineer at the University of Technology, Sydney, Australia. And he's actually crossing over from academia into industry. He's working on a medical company called Neogenics Biosciences. And basically, he's going to share with us his PhD work today that has now translated or is on the way to translating to this technology for assisted reproduction. And he's now working closely with IVF clinics on this technology. They also use AI to help with the translation of the technology as well. So without further ado, Steve, go ahead.
Awesome. Thank you, Angela. And thank you, Cindy, for the introduction. I'll go ahead and share my screen. Can you guys see that okay? Yep. Fantastic. And I'll just move you guys out of the way there. Awesome. This is very exciting. Thank you for the introduction. Thank you for inviting me to speak today. So my name's Stephen. I've recently finished my PhD at the University of Technology, Sydney, and as Angela mentioned, developing microfluidic tools and some software for selecting sperm in assisted reproduction. So before I start, I'd like to mention small conflict of interest. I guess maybe not even, a lot of this data predates the startup, but worth mentioning that I have started a company called Neogenics, which is spun out from my PhD, and we're busy commercializing modified versions of what I'm going to show you today. And but if you have any questions, feel free to reach out to our lab or reach out to me, especially if you have questions about commercialization or technology. So today I'll take you through a little bit of what is infertility to start with, and male infertility in particular, and the importance of selecting good sperm for assisted reproduction. Then I'll take you through our microfluidic device, what we've developed in this couple of studies that we've done during my PhD, and what's next for us. So 1 in every 6 couples struggle with infertility. 1 in every 6. And while we now have assisted reproduction or IVF to help treat infertility, a lot of people don't realize that about 78% of IVF cycles fail. 78%, that's a lot. Another concerning fact is that not only is fertility declining across the board, but male infertility in particular has taken a hit. Sperm counts have dropped by about 50% in the last 40 years. And 30% of all infertility is just male factor infertility. 55% of the time, there's a male and female factor contributing together to cause a couple to become infertile. So male infertility plays a big role in infertility and infertility management. Although, like I mentioned, we have assisted reproduction, of which there are 3 main types, 3 main types of fertilization, basically. And you don't need to know too many details about these. They're mainly administering different ways of fertilization. So we can either inject prepared sperm into the uterus, we can surround an egg with a population of sperm to recognize and bind to the sperm and then fertilize the egg, or we can inject a single sperm into a single egg, which is called ICSI. The common theme between all of these, though, is that you require sperm selection, regardless of the type of infertility or the modality of treatment that we're providing. The number of sperm we need changes depending on the treatment we're providing, but you still need to do sperm preparation and sperm selection. So what is a good sperm, though? The WHO defines healthy sperm by their motility, how fast they're swimming, their velocity profiles, the straightness and curvature of the sperm's motion, their morphology. And we can also define sperm quality by DNA integrity or DNA fragmentation, DNA damage. literally physical fragmentation of the DNA within the sperm, and that's correlated to lower embryo quality. It affects miscarriage rates as well. But the goal here, and the goal for my PhD developing devices, is to select competent, mature, and undamaged sperm for assisted reproduction. So the problem that we have in clinics is conventional sperm selection involves the centrifugation of sperm at around 500 Gs in a silica particle buffer. We call it density gradient centrifugation. Or there's a process called swim up, which also involves centrifugation as well, and involves some washing steps. So when I'm pitching it, when I'm talking, more generally, I always say, as a relative, hypergravity for an astronaut is like 3 Gs. Fighter pilots pull about 9 Gs. And we're spinning sperm at 500 Gs. So you can imagine that these sperm aren't having a particularly great time, and they have no way to repair themselves. And what this can do is this process can cause DNA damage in the sperm. It's also an extremely manual, unstandardized process. It's prone to a lot of human error. And the flow-on effects for the treatment is if you're using sperm with damaged DNA, you can get results of poorer embryo quality, lower implantation rates, so the embryo we grow doesn't stick to the uterus, basically, and it can cause higher rates of miscarriage. And depending on what type of assisted reproduction you're doing, you can have anywhere between 2.5-fold increase in the risk of miscarriage to a 5-fold increased risk of miscarriage, according to clinical studies, when you're doing IUI, where you're injecting sperm into the uterus. So the quality of the sperm when we're fertilizing the egg and making an embryo matters a lot. So what we've gone and done at UTS with the Varkiani lab in collaboration with Virtus Health and IVF Australia is we've developed a multi-stage sperm selection device able to wash and select sperm based on their motility. So not only does the device select sperm by providing guidance similar to the grooves of the female reproductive tract, it also resuspends sperm in a nutrient buffer. Semen becomes toxic over time. It's not meant to be outside the body, so we want to get the sperm out of the semen and into a buffer as quickly as possible. We do that by creating a fluid boundary between the semen, which you can see D over the edge here, and these microfluidic channels, which we've pre-filled with our buffer, and that has all the sugars and antioxidants and everything that's going to keep a sperm happy and healthy. It's kind of like simulating the junction between the vagina and the cervix, where the sperm move out of the semen into the female fluids. And then after that, it basically becomes kind of like a sperm Olympics, where human sperm have a unique ability to sense the geometry around them and follow boundaries, follow corners. And the ones that can do that and exhibit that behavior, we can collect those, we keep those sperm, and they can outcompete the ones that don't perform that well. We also developed an extra filter as part of this study for filtering out apoptotic sperm using paramagnetic beads and trapping sperm coated with annexin-coated particles. But that's just an optional inclusion. The main driver here is motility for the primary selection and filtration. And the way that we did this, and we made these geometries, is we used 3D printing initially to replicate the numerous grooves of the female reproductive tract to help guide and wash these sperm. So you can see some cross sections here of different stages of the device. Basically, it becomes like a multi-layered highway for sperm to traverse through. But it gets more and more complex as it goes, as the sperm traverse through the device, and they have to outcompete themselves. Then there's features at the center of the device to trap the good ones there so they don't swim back the way they came. But after this process, essentially, you've got a healthy population of sperm that's ready to be used for fertilization. So here's one I've prepared earlier. Funnily enough, no one's ever asked for a live demo. I can't imagine why. But on the left here, you could see raw semen, and you can see quite a lot of contamination in the background. Here's quite a terrible sample, actually, with lots of epithelial cells, white blood cells, red blood cells, dead sperm, dying sperm, and lots of particulates and whatnot. All of that has a toxic buildup over time. But after the device, you can see we've got really highly motile population of sperm in a buffer ready to go to be used for fertilization. And the process of our device, it takes about 1/3 of the time as most conventional methods. The conventional methods do vary a lot, though, from clinic to clinic. But it's also a lot simpler. And what we noticed, especially, which I'll mention a little bit talking about working with clinics, is a lot less error with our process, where the device basically is you inject the device, you prime the device with a buffer, you load your semen, you wait 15 minutes, and then you collect your semen, as opposed to conventional processes where you're aspirating sperm, washing sperm, centrifuging sperm, repeating, and so on and so forth. So we've run 3 studies to date. First one was with healthy donors from the general public at UTS, where we compared one of the conventional methods here, swim up, SU, against raw semen against the device. And we saw a massive reduction in DNA fragmentation, or that DNA damage. And the exciting thing is that it doesn't really matter what we started with. Infertility is a gradient. Some men come in with higher levels than others, but we're always getting it down to close to 0. They're averaging around 1.92% DNA fragmentation, which is fantastic. Obviously, we're being more selective than current methods, so our concentration takes a bit of a hit, but you can incubate the device for a little bit longer. We're still getting enough numbers to do all those different types of assisted reproduction that I mentioned earlier. Of course, the vitality, we measured the vitality. We wanted to make sure that the 3D printed device wasn't having any toxic buildup over time. We actually kept incubating the sperm post-selection for a couple of hours and didn't see any adverse effects. We also looked at the motility of the sperm compared to the conventional methods, and we're averaging really, really high grade A, what we call progressive motility, which is where the sperm have a good forward motion. They're not kind of just sitting there twitching. We also looked at the velocity profiles of the sperm in our device with heat and our device without heat. Typically, we want to do sperm selection with heat because it hyperactivates the sperm. Their chemistry changes, they what we call capacitate, and they start burning more energy and swim faster. So you'll get more sperm through the device with heat, but we saw higher velocity profiles in general from our device, which is great. We also looked at the cryo recovery. Conscious for time here, so I'll go through this quickly, but we saw slightly better sperm recovery from cryopreservation versus conventional methods. If we pre-sorted and cryopreserved our sperm, we saw better recovery afterwards. Cryopreservation for sperm is quite harsh. About 50% on average of the sperm die just from the freezing process. So our sperm were a little bit more resilient to that freezing process, which was good, but we do need to do a little bit more experiments here. Number is a bit low. Then the next step for us is we moved to clinics. So we gave these devices, we made some slight geometric changes, and then we moved to testing at Melbourne IVF and IVF Australia and Monash Hospital in the EPRD. So we ran 2 separate studies. The first one here is at, this one's at Monash Hospital. Similar sort of trend, which is fantastic. Now these are samples coming through for diagnostics for their IVF treatments, and we're still seeing a fantastic reduction in DNA damage, regardless of what the starting level is. Progressive motility was terrific. And then we continued on and we did another test at Melbourne IVF. Primarily, we've also done testing at IVF Australia. We're still doing testing actually at IVF Australia and Melbourne IVF, but this was in David Gardner's lab, where we saw a dramatic reduction again in DNA fragmentation, which is terrific. Again, all used by trained embryologists. We tried to get a spread of embryologists, different people, senior, junior embryologists across a wide variety of sample qualities. But it's great that we're still seeing high motility. We're still seeing high reduction in DNA fragmentation. So next steps for us now is we're moving towards manufacturing, clinical trials. We're preparing our AI modules to pair with these devices as an extra layer of filtration, where you can debulk and select your healthy sperm from this device, then provide another layer of assessment through AI. But I think I'll end it there. Thank you very much, and happy to take any questions. And obviously, I'd like to thank the clinics that we work with, of course, IVF Australia, Melbourne IVF, Virtus Health have been fantastic. AMP has been fantastic, providing manufacturing capability for us, A&B Labs, Reza Nosrati's lab there, and of course, Varkiani Lab, which is our lab out at UTS. And this is Majid here, who's the head of the lab, if you'd like to talk to him about microfluidic technology.
Awesome. Thank you,
Cool.
Steve. So I think we'll leave the Q&A all the way until the end. So Steve, if you could stick around until the end, that would be great. Fantastic. Okay, so after that great talk to kick us off, we have our speaker for today for the longer talk, and that is going to be Hirofumi Shintaku. He is a professor at the Institute of Life and Medical Sciences at Kyoto University, and also a Riken Hakubi team leader. So the Riken Hakubi team leader position is actually very prestigious in Japan, if I understand correctly. And he's also received many other awards, including the Young Engineers Award from the Japan Society of Mechanical Engineers, which is the highest honor for young mechanical engineers over there. His current research is focusing at the sort of nexus of technology development and single cell biology, and leveraging electrokinetics in micro and nanoscale, and sort of integrating that technology to understand cell biology and sometimes also disease. So I know Hiro has his own introduction for you guys, so I'll let him tell you about his work.
Thank you, Angela. Can you hear me?
Yes, yes.
Okay, that's great. Thank you for kind introduction, and I'm so excited to be here to introduce our research work. Yeah. Right. Yeah, so before we dive into the research, I would like to show the acknowledgement slide. Currently, I'm directing two laboratories at Riken and Kyoto. I've just started my new laboratory at Kyoto University this last year, April, and moving from Riken to the Kyoto. And most of the work I share with you today has been done in my lab at Riken. I would like to acknowledge the hard work done by my team members and collaborators, as well as the funding source that made that work happen. Right. So as I am currently working at two sites in Japan, I'm traveling a lot with a bullet train called Shinkansen, but this is relatively comfortable. Riken is located in the Tokyo region around here, and Kyoto is in Kyoto and the Asian capital. It takes about 2.5 hours. It's a nice and comfortable traveling with the bullet train. So if you have a chance to come to Japan, visit me either at Riken or Kyoto. If you visit both sites, you can enjoy the nice view of Mount Fuji from the bullet train. So this is a picture I took some day before. Yeah, I like to advertise my new place, Kyoto. Kyoto is a very beautiful city. You can see many Asian traditional shrine and a temple, and you can visit many historical places and enjoy nice food. So if you come to Kyoto, I am happy to treat you a nice Japanese food. And my lab in Kyoto is very spacey. It is double size compared to that weekend. So now we are working hard to build up the new laboratory to push our science forward. And we are also hiring. So if you are interested in coming to Japan and working in Japan, doing research in Japan, just shoot me an email. And all right, so today we'd like to discuss about our research on electroporation, nano or micro electroporation. We are leveraging nano or micro electroporation to expand a single cell omics. So our group utilized micro nano electroporation and electrokinetics to expand the capability of a single cell mix in terms of resolution, temporal, and phenotyping. What I mean by the micro nano electroporation is that we manipulate the electric field in micro or nanometer scale with micro-nano fabricated structure. In a conventional electroporation, we basically use uniform electric field to permeate cerebral membrane. But in micro-nanometer nano electroporation, we use focused electric field in nanometer or micrometer scale to do something interesting. And as you know, the single cell RNA sequencing is the most powerful approach to profile gene expression of single cells or cell state, cell type at large scale. And the microfluidics has contributed to significantly to make the single cell RNA sequencing approach high throughput. Now we can study complex biological phenomena involving multiple individual cells. However, one caveat here is that the single cell sequencing approaches basically correct snapshots of single cell. So we basically can know the gene expression at the single time point, but we cannot track the cellular dynamics after the measurement. So we basically kill the cell at the time point of the measurement. To overcome such problem, many researchers have proposed computational approaches to infer the dynamics of cells by smoothly connecting single-cell RNA-seq data. One famous approach is pseudo-time analysis, or utilize internal grids to estimate the RNA velocity and so on. Our group has also proposed a unique approach, so-called SyncSeq, that leverages the difference in the nuclear RNA cytoplasmic gene expressions to infer the dynamics of cells. But these are still inference of dynamics of cells, so we just need a real measurement tool for cellular dynamics. To measure the dynamics of a cell, we are working to develop a new method that allows to measure the gene expression as time series. I would like to discuss a little bit later today. But before discuss about this new approach, I'd like to spend some time to discuss about our SyncSeq approach. Right, so in SyncSeq approach, we use microscale electroporation. So in our SyncSeq approach, we focus the RNA trajectory in eukaryotic cell. Eukaryotic cells basically transcribe RNA in the nucleus and then transport RNA molecule to cytosol to translate them to proteins. According to this cascade, we hypothesize that the nuclear RNA and the cytoplasmic RNA may have information at different time points. So what we thought is shown in this slide, that nuclear RNA tells us the future state of the cell, while the cytoplasmic RNA does current state. So taking the difference between these two gene expression, we can probably infer the dynamics of the cell. To measure such difference between nuclear and cytoplasmic gene expressions, we developed a method called the SyncSeq, single-cell integrated nuclear and cytoplasmic RNA sequencing. So what we do is basically we physically fractionate nuclear and cytoplasmic RNA, and then we do sequencing on those two subcellular RNA independently. So in order to do such measurement, we need to fractionate the tiny amount of RNA. As you know, as you can imagine, the amount of the RNA in a single cell is really tiny. It's in the order of 10 picogram, 10 to the minus 12. So that's really difficult to fractionate cytoplasmic RNA, fractionating cytoplasmic RNA versus nucleus. That's where the microfluidics comes in. So our microfluidics system for SyncSeq is shown in the slide. So it has a simple geometry, T-shaped channel with a hydrodynamic trap over here. Our system uniquely uses electric field for selective lysing of the plasma membrane while keeping the nuclear membrane intact and extracting cytoplasmic RNA from the cell. So in our protocol, first we capture a single cell at a hydrodynamic trap, and then we just apply DC electric field across the channel. The DC electric field induces ionic current going through the microfluidic channel. At this hydrodynamic trap, all the ionic current going through the gap, a tiny gap, microscale tiny gap over here. So that creates focused electric field. And we leverage this focused electric field to selectively lyse the plasma membrane and effectively extract the cytoplasmic component from the lysosomal cell. And we transport the extracted cytoplasmic component to downstream by electrophoresis or iStack-phoresis. And when the cytoplasmic component arrives the downstream reservoir, we deactivate electric field and we extract the cytoplasmic RNA from downstream and nucleus from upstream. And then we do off-chip library preparation for the next-gen sequencing. Now I want to show you how this system works in the movie over here. So what you see here is, this ball is a single cell captured at a hydrodynamic trap. Hydrodynamic trap is invisible in this movie, but it's on the right side of the cell. And by applying electric field, lysis happens within 1 second and the extraction completes within 20 seconds. So the extraction, lysis and extraction is really quick. That completes within 1 minute. And coupling with off-chip prep and the next-gen sequencing, we can quantitatively measure the gene expression in subcellular compartments, the nucleus and the cytoplasm. We can tell which genes are enriched in cytoplasm or nucleus. And also by coupling with long-read sequencing, such as oxygen nanopore or Oxford nanopore technology, we can also study structural difference in cytoplasmic and nuclear RNA. So these are the same genes detected in nucleus or cytoplasm. As you can see here, these genes are having a different structure. So nuclear RNAs are less mature than the cytoplasmic RNA. So, all right, so as we introduced, the sync-seq is called the cool microfluidics, but it's still an endpoint. We kill the cell at the time point of the measurement. We just wanted to invent another new approach that can truly track the gene expression of living cell as time series without killing the cell. So we move on to the second topic. So to do that kind of gene expression analysis time series, we utilize nanoscale, nanometer scale electroporation, and we expand the capability in temporal direction. So our basic idea is that we sample a tiny amount of RNA to keep the cells viable. And what we do is that we use a conventional glass capillary with a nanometer scale inner diameter. So the approach is very, easy. So we use this glass capillary with nanometer scale inner diameter, and we apply a tiny amount of the voltage to detect the touching on the surface of the cell. So as we approach to the cell surface with this nanometer inner diameter glass capillary, we can detect the touching by sensing the resistivity change. And after sensing touching on the surface of the cellular membrane, we apply optimized biased voltage to transiently create pore in the plasma membrane and extract cytoplasm, a tiny amount of cytoplasm, electrokinetically without killing the cell. And Kyoto Tori of our postdoc in my group spent some time to optimize this process, and he found the very nice buffer condition and applied voltage and the size of the pore, and to balance between the extraction efficiency and cellular viability. And so now I want to show you how we do this extraction. So these are cells stained with the calcine, and now we have a glass capillary touching on the surface of this cell. And by applying a this biased electric field, we can extract the cytoplasm. So probably you can see that the fluorescence intensity of this cell decreased as we do extraction. And using this nano electrokinetic sampling, we demonstrated extracting RNA with keeping the cells alive and performed the RNA-seq with a tiny amount of RNA. So here we apply this sampling approach to HeLa cells with Fuji fluorescence protein system. The Fuji is a fluorescence protein system which indicates the cell cycle progression with the fluorescent protein. So starting from the red color to the green, so by detecting the color of the fluorescent protein, we can tell where the cell cycle phase is. And we here did sampling every 2.5 hour, 0 hour, 2.5, 5 hour, and 7.5 hours. And so four time points. And we track the cellular dynamics or cell cycle progression. And here we compare the cell cycle progression between control and sample, and we didn't see any perturbation in the speed of the cell cycle progression. And every sampling, we sample about 0.6 picogram of RNA. That's only probably about 3% of the total RNA of the cell. And we use this 0.6 picogram of RNA to do RNA sequencing. And with that tiny amount of RNA, we detect 10,000 transcripts per sampling. And we detect 2,000 genes per sampling. So we can track the gene expression as time series at a 2.5 hour time resolution, temporal resolution. Using that gene expression time series data, we can draw the trajectory of the individual cell. So here we show the data taken at a different time point, presented in the UMAP using the gene expression data. As you can see here, the UMAP clearly show how the cell cycle progresses. And also by looking at the gene expression of individual genes related to the cell cycle, we can see different going up and down behavior. Now with our unique time series gene expression data, we can compute the transcriptional kinetics, synthesis rate, and degradation rate. So these variables are very important to infer the dynamical behavior of the cell. So as we can keep the cells alive, we can measure the gene expression time series. And also we can monitor the behavior of the cell using optical fluorescence microscope. So we can combine optical microscopic data and a gene expression time series. And here we investigated the perturbation or fluctuation in the cell cycle progression. So individual cells show very different the speed, velocity in the cell cycle progression. And we try to explain if we can explain that fluctuation using our data set. And we found that the transcription rate, which we compute from the gene expression data time series gene expression data, can explain that fluctuation precisely. We next applied our nanoelectrokinetic approach to study embryogenesis of C. elegans. We call this approach as live organismal transcriptomics because we can track the gene expression of organism. Here we applied our nanoelectrokinetic sampling at single cell stage to full cell stage, and we monitor whether this embryo hatch or die. Now I want to show you how we do sampling of surpassing of embryonic cell. So this is embryo at single cell stage, and this shadow is showing the grass capillary touching on the surface of the embryo. And we sample by applying electric field, and the cell divides into two. Then again, we sample those two cells, and again, we wait until cells divide. Now it's full cell stage, and we sample. Okay, so this embryo is sample, and another one sitting next to it is a control. Oh, so I want to show you this, my favorite movie. Oh, oh, oh. So all right. So this is my favorite movie, so I need to show. So we monitor whether this embryo hatch or die, and control one hatch, and also sample one hatches. So in order to do a little bit of biology, here we applied our approach to the OMA1 mutant. OMA1 protein inhibits the transcription and translation. Interestingly, OMA1 mutant dies during embryogenesis at about 50% chance under restricted temperature condition. The embryonic fate of hatch or die is apparently stochastic. We thus applied our nano-electrokinetic sampling to measure the transient gene expression at multiple cell stages and then observe their embryonic fate to find out the mechanistic explanation for the apparently stochastic process. In this experiment, we obtained a gene expression from 23 embryos that correspond to 115 single cells. In a single cell RNA-seq or standard single cell RNA-seq, we typically create the data matrix as gene in vertical direction and cells in horizontal. But as we in this experiment, we know that which cell comes from which embryo. So instead of placing the data in this direction, we concatenated the gene expression in the vertical direction. And we created gene expression of single organism. In addition, to implement the time evolution of the gene expression in the data, we computed real velocity of the gene by taking the difference in the gene expression along the cell lineage, the VAB and VABA. So these are velocity from single cell stage and two cell stage and four cell stage. And we obtain another velocity, a P cell lineage as well. So VP1, VP2. So these are showing how much gene expression change created during the cell division. So we concatenated these four velocity to the gene expression data. Further, to implement the relation between the two cell lineages, P lineage and AB lineage, we computed the velocity difference between the two lineages. Taking the difference between the VAB and VP1, we obtain this variable, delta. So we call this divergence. So by calculating the velocity difference, we basically obtain the gene expression difference between these two cells. And we also computed the velocity difference from of those two velocities. So these are velocity computed from two cell stage and a four cell stage. And by calculating the delta, the velocity difference from two to four cell stage, we obtain this variable. So this is basically the first term of this is basically the gene expression difference between these two cells minus delta ABP1. So this is showing how much divergence was created from two to four cell stage. So we concatenated these two divergence into the data set. And we then explored which variable is the most responsible to explain the embryonic fate, expression, velocity, or divergence. And to make the long story short, we found that the divergence, especially from 1 to 2 cell stage, was most responsible to explain the fate of the embryo, hatch or dead. And we further explored the determinant gene at the one cell stage, leveraging the divergence, and we identified FZY1 gene is most responsible to explain the embryonic fate of OML1 mutant. We further found that FZ11 located in the center of the correlation network of gene expression. So that means the FZ11 was very important determinant to determine the embryonic fate. So these data support that our ribo-organismal transcriptomics is very powerful in identifying the determinant gene and obtaining the mechanistic insight for the apparently stochastic fate determination. Right, now I'd like to move on to the last topic, nano-electrokinetics for phenotyping. So we named this our approach as elastomics, which measures the mechanophenotype as well as the gene expression at single cell resolution. Mechanophenotype is an important cellular characteristic. It is involved in many biological contexts. For instance, the cell division, which is most fundamental process. The cell, mechanical phenotype is regulated, properly regulated, and cell migration. So when the cell migrates, the mechanical property is very important. And the tissue development or morphological change is also involving mechanical property. However, at this moment, there are no single-cell approaches to profile the mechanical state and the omics layer simultaneously at single-cell resolution at high throughput. So this situation motivated us to develop a new approach. So here we focus the cell surface tension, which is one of the important mechanical properties of cell. The cell surface tension is approximately sum of the plasma membrane tension and cortical tension. To develop the approach to measure the cell surface tension, we focused on the change of the free energy during the pore formation in the lipid bilayer by via nanopore electroporation. The change of the free energy is a function of radius of pore, the surface tension, and edge tension. So this equation tells that the larger pores are created in the lipid bilayer with high surface tension. This means that if we can profile the cell surface tension by measuring the radius of the pore created by electroporation. So on the basis of this idea, we developed elastomics. In elastomics, we see the cells on the tract edge membrane, which is a non-conductive film with a bunch of nanoscale pores. So these are, SEM image showing the cell and also nanoscale pores. So we basically see the cell on top of the tract edge membrane, and then we apply electric field across the membrane. As this tract edge membrane is made of a non-conductive material, all the ionic current goes through the nanopores. That creates nanoscale focused electric field. And that focused electric field is excited on the plasma membrane of the cell. Because the size of the pore, so that non-focused electric field creates pores, tiny pores, transiently, tiny pores in the lipid bilayer. In the meantime, we deliver molecules into the cell. As the size of the pore, radius of the pore created by this approach is dependent on the cell surface tension. The size of the molecules uptaken by the cell are dependent on the cell surface tension. The cells with low surface tension uptake only smaller size of the molecule or smaller amount of the molecule. But the cells with high cell surface tension creates larger size of pore that uptake more molecule or larger size of molecule. And we wanted to quantify how many molecules or how large molecules are delivered into the cell with this method by sequencing. So we developed DNA tagged dextran, we call DTD, using dextran molecule with different molecular weight. So we prepared a DTD with a different dextran molecule with different molecular weight. And we label those dextran molecule with DNA tag. So DNA tag has a unique barcode corresponding to the size of the dextran molecule. And we add poly-A sequence to this DNA tag in order to do sequencing simultaneously with transcripts. So in this process, we deliver this DTD into the cell just by applying electric field. And then we transfer the treated cell to standard single-cell RNA sequencing approach. Here we use 10x genomics Chromium system. As DTD has poly-A sequence, just by doing RNA-seq, we can quantify the gene expression as well as the DTD. And we obtain the gene expression from mRNA and cell surface tension from DTD. So we apply our approach to different type of cell. So here we are showing the number of DTD delivered into the cell. As you can see, the smaller size molecules are delivered more than the larger size molecule. And on the right figure, to examine if the elastomers can probe the self-surface tension, we applied our approach to MCF10A treated with cytokeratin D to bring down the self-surface tension. As you can see here, the MCF10A cell treated with cytokeratin D shows smaller amount of DTD. So this means that the elastomers can probe the reduction of the self-surface tension. Next, we applied our approach to various cancer cells with different malignancies. Actually, highly malignant cells are known to have low self-surface tension. Here we show that the PC3 and MDA MB231 cells, which are metastatic cancer cells, uptake a smaller amount of DTD. So green colors are showing the molecule we deliver by nanoelectroporation. So those highly malignant cells have low surface tension. Therefore, they uptake small amount of molecule by our approach. On the other hand, less invasive cancer cell line, MCF7, uptake more molecule by approach. And non-cancerous cell line, MCF10A, uptake more. And with elastomers, we can measure not only the cell surface tension, but also gene expression. So by integrating gene expression data and DTD counting, we can identify the cell type from the gene expression. So this is showing the UMAP data of individual single cell. So we can see these different cell types are clearly segregated in UMAP space. And overlaying the DTD count, we can see which cell type has higher or lower cell surface tension. So here the MCF10A cell has the highest cell surface tension. Next, we show application of elastomers to several senescence. Cell aging due to the replication cause increase in the cell surface tension. This increase in the cell surface tension may be involved in the age-related diseases. But the molecular mechanism behind the change in the cell surface tension and its cellular heterogeneity remain explored. Therefore, we applied our elastomers approach. To explore the cell surface tension along the cellular senescence, we here applied elastomerics to TIG1 cell, which shows replicative senescence. As we expected, our approach delivered more DTD to aged TIG1 cell. So this is the aged one and relatively young one. And we observe that the aged one uptake more DTD. And that is also detected by sequencing. So aged one uptake more DTD. Implying the cell surface tension increase around the cellular senescence. By leveraging the correlation between the more amount of the delivered DTD and the gene expression, we identified genes that may be involved in the mechanophenotypic change along the cellular senescence. As we expected, we observed that genes upregulated around the cellular senescence shows positive correlation. So these are genes are showing the positive correlation, and these are related to the cellular senescence. On the other hand, the genes downregulated along the cellular senescence shows a negative correlation down here. To identify the regulatory gene, we here applied multiple regression analysis, and we identified RRAD that showed a high contribution to the cell surface tension. The RRAD is known as a tumor repressor, which represses glycolysis mainly through an inhibition of GLUT1 translocation to inhibit the import of glucose. To confirm the relationship between RRAD and cell surface tension, we analyzed the glycolysis pathway. First, we performed siRNA assay to bring down the RRAD expression, and siRNA assay dramatically reduced the cell surface tension, as you see here. Second, we inhibit the glycolysis by using the 2-deoxyglucose assay, and that increased the cell surface tension in TIG1 cell. So this is showing the increase of the cell surface tension. So these results indicate that RRAD is involved in the regulation of cell surface tension along the cellular senescence via glycolysis axis. All right, so today we introduced our approaches that leverage microscale or nanoscale focused electric field to exploit the new functionality of the electroporation. I hope you could enjoy at least one of those. And lastly, showing off our group picture. I'd like to stop my talk, and I'm happy to take any question from the audience.
Okay, awesome. That's really good timing as well. So if there's anybody who wishes to ask questions, you can either put them in the chat box, or you can also raise your hand to go on live. So let me first, okay, and Stephen, are you also going to come back for, I think we're just going to do the Q&As together. Yeah, awesome. So we can start first with some of the questions that are in the chat box. So we'll start with those for Stephen. So there's a question from Eric. If I heard correctly, sorting depends on sperm sensitivity to the edges of your device. Is this correct? And does this edge detection pathway differ for other cells like macrophages or, etc.?
Thanks, Eric. Yeah, that's a good question. Yes, correct. The sperm migration in our device is dependent on edge detection. The way the devices are currently orientated, it changes throughout the device, but sperm, they are quite unique compared to macrophages. It's not kind of like, it's not more much of an amoeboid or even the same sort of ciliated sort of movement. Sperm exhibit a suite of different swimming patterns. They'll be either helical or chiral sort of ribbons. But what we find in these devices, when sperm are confined in their geometry, they'll enter kind of like a slither swimming mode. So the head of a sperm is sort of a disc shape. They'll settle mostly by gravity as they go through our devices. When they hit the floor, they tend to stay there. And then they end up staying in that mode, and then they start exhibit the boundary following behavior. And they kind of end up bumping continuously into the wall, and the waveform of their tail changes when they're in this position. And what we've found is that you can change the angle. If the channel is straight, they'll stay straight. But if you change the angle, you give sperm a turn, then only sperm with a decent enough head morphology who can will keep that angle and actually be able to turn corners. And there's a correlation between that and the DNA integrity of the sperm, which is kind of what we're exploiting. But there's a lot still going on, people trying to model sperm motility and sperm behavior. So there's a lot to be done. I hope that answers your question.
Okay, great. There's another question from Sai. So thank you for the great talk. It was very interesting to hear about the process of translating the research. What do you anticipate are some of the challenges to the wide adoption of this product, and are there any regulatory challenges?
Yes, there's a lot of regulatory challenges. I think the first big takeaway is whatever you design in, the biggest learning is whatever you design in academia as part of your PhD or part of your research is not what's going to make it into industry. No matter how close you think you are, 99.9% of the time, it's going to have to change, and you're going to have to do a lot of user validation to appropriate your product to be adoptable in the clinic. There are a lot of regulatory hurdles, so regulatory strategy is really important. We've seen a lot of changes recently in the regulatory pathway. The general trend across the globe at the moment is upregulation. So we have the latest MDR. We've gone from MDD to MDR in Europe. The rest of the world's kind of following suit. So it's a big upregulation. So that means more clinical data. Lots of companies that are already out there are still having to even revalidate to upregulate their devices. But we are able to leverage some similar in class. There are other people who are trying to do other, mechanisms, but similar materials or similar sort of microfluidic techniques for sperm selection. So that lowers the barrier to entry a little bit, but regulatory strategy matters a lot. And I think working closely with clinics and the end users matters a lot. Get everything into their hands quickly, because you're going to have to appropriate your technology to fit the workflow, for us, the workflow of IVF.
Okay, the next question is, did you do any phenotype testing of the sorted sperm, like how the design of the device affects the phenotype function? And maybe my own add-on to that is, were there any animal experiments or I don't know, any kind of experiments that actually show improved outcome, or is that like part of the company and you can't talk about it here?
Yeah, there's a little bit of confidentiality here, but yes, we've done animal stuff. We actually started in bull sperm because they're just better. They're just easier to work with than human sperm. So they were a good model for testing microfluidic sperm selection, and then moving to human semen, which is generally, a bit messier. We haven't done any embryo studies yet, but that's what we're doing now with Neogenics, is trying to get some mouse model workup done. It's a bit hard to generate infertile mouse models. Mouse models are very fertile just in general. But yes, in terms of phenotyping, we've got some unpublished data that we're working on now, looking at the different proteomic profiles, different surface markers of the sperm selected by different microfluidic channels. And then we have optimized our channels to try and get the best reduction in DNA damage and the highest motility characteristics. We characterize motility in 4 or 5 different velocity parameters. But similar to Eric's question, we can moderate the channels, their height and thickness, and the curvature that we get in our channels, also in the third dimension as well, to make sure we're being more selective as the device progresses, or as the sperm progress through the device.
Got it. Cool. It's cool to hear that you guys are doing animal study. That's always challenging.
Oh, yeah.
Yeah. So the next couple questions are for Hiro. Do you use active suction or capillary forces to aspirate cytoplasm from the cell? And I think later on there was a question about, what volume of, what volumes are you sucking out and how do you control that?
Yeah, so that's a great question. So we actually don't do any aspiration or suction. We basically apply just the electric field, and our voltage bias is like biphasic. So we basically induce we don't transport the solution, but we transport only the charge molecule. So we don't see any volumetric change of the cell before and after the sampling.
Oh, that's interesting. Okay, I did not that was not something I was aware of. That's pretty cool. Okay, for the next question, Hiro, for the DC-based electroporation, what is the material of the electrode and the dimensions? And does your lab observe electrolysis in the devices during electroporation?
Oh, that's also a very important technical question. We use a platinum electrode, but usually we place those electrodes very far from the cell. So actually, at the surface of the electrode, electrolysis is happening, and maybe the pH is a little bit changing, but though that is happening very far from the cell, so we don't see any influence to the cell from the electrolysis.
Mm, okay. And then, the question after that one from Fernando is, how long is the voltage pulse applied for the electroporation? And does the nanopore reseal after the pulse, or does the pore remain open for some time?
Yeah, that's a good point. Well, actually, I said DC, but DC, the voltage is DC, but it's pulse voltage. So it's like a millisecond scale. Well, nano, in the case of elasmics, we apply like 10 millisecond pulse. But for the electrokinetic sampling case, we apply 100 millisecond scale pulse. So it's a sub-second pulse.
Okay. And does the pore reseal, or do you think it remains
Oh,
open for?
yeah, that reseals. So we haven't observed, but we try to keep the invasiveness as small as possible. And we see they are living for some time because therefore we think the creative pore reseals.
Mm. Okay.
We,
And
we,
then,
we don't know the time scale exactly
yeah,
because we haven't measured, but it should be very quick
okay.
process.
Okay. There's a question about the electrokinetic sampling. Like, what's the minimum volume of RNA that you need to do the downstream analysis? I guess the RNA-seq.
Yeah, so if we want to do deep sequencing, we want to take more. But at this moment, we balance between the sensitivity and cellular viability. And we are now comfortable with extracting about 1 picogram of RNA. But it depends on which sample. Embryonic cell are usually relatively large than somatic cell. So maybe we can sample more RNA. But we haven't tried. We always try to find the sweet spot where we can keep the cellular viability and the sensitivity.
Yeah, yeah. Yeah, 1 picogram actually seems like quite a lot. It's 1/6. Okay. There's a question about the sync-seq. So how small is the size of the constriction channel, and what is the average ratio between the channel size and the cell size?
Yeah, the size of the gap was 2.5 micrometer. And the cell size was like 10 micrometer. So it's about one-fourth.
Okay, and I have a question about the nanoporation. So I guess people have been using electroporation to do sort of like delivery, drug delivery and stuff. And I think what's interesting about your system is that it also has this cell response to the surface it's on kind of component. Have you guys studied, you mentioned like you looked at different cancer cells, right? And it has different elasticity. So have you studied whether, in the case of drug delivery, that would actually affect or impact the size of the molecules that you can deliver effectively into a cell or something like this? Like whether the cell's elasticity or, whatever their biomechanics are actually affects the efficiency of delivering a certain drug because of the size or something like that.
Yeah, that's a very good question. We actually deliver dextran molecule, which is not functional, but we want to deliver also the functional molecule as well, and we want to monitor the behavior or response to the delivered molecule of the cells. That's our future work, and we hope that maybe we can deliver molecules selectively. Maybe we want to deliver drug to malignant cell, but unfortunately, the malignant cells have had low surface tension, so they uptake less amount of drug or less amount of molecule. So unfortunately, our method is not so effective to kill like a highly malignant cell. But maybe in some cases, some dangerous cell have high surface tension. In that case, maybe our approach is very effective to selectively kill those dangerous cells.
Right, right. Well, I think it's good. I mean, it's important also to know that fundamental relationship, because then if somebody's, let's say, designing, the delivery vehicle, they might want to restrict it to a certain size to account for that. So yeah, for sure, I think it offers a very important information. Okay, are there any further questions? I don't see any more questions in the chat box.
Yeah.
I have a question.
Oh, Cindy.
Sponsor.
Yes.
Yeah. So
You need to
thank
raise
you.
your
Yeah.
hand, Cindy.
Oh yeah, sorry, I should have done that. Thank you, Steve and Hiro, for excellent talk. So I have a question for both of you. Maybe I'll ask Hiro first, because it's along the line of the surface tension. I wonder if you have, if there's any confining effect of the viscosity of the cytoplasm in the uptake of molecules, because like surface tension control the size of the hole, right? But presumably the viscosity of the cytoplasm would also control like how fast the molecules can get in. Like, have you seen any confounding factors, or this is even like a concern that like viscosity might change significantly from one cell type to the other?
Yeah, at this moment we have tried up to 500 kilodalton dextran, and so that dextran molecule diffuse relatively quickly within the cell, and they are uniform within the cell. So at this moment we have not seen any confinement or localization, but we hope to develop such molecule which shows localization, because if we can have such molecule, we can probably measure the distribution of cell surface tension. So at this moment, we just measure like average cell surface tension, the single value from a single cell. But if we can localize or confine the molecule in a local space, we can probably measure the local cell surface tension. Yeah, that's a great point.
Yeah, that's very cool. Thank you. And Steve, I have a question for you. I was pretty struck by your comment about how like when you translate a technology from the lab to industry, like no matter what you do, like what the product will look like would be so different than what you develop in your PhD. Do you have, so like there are a lot of students here in the audience who are interested in translating that technology. What recommendation or advice do you have for the students who are thinking about bringing what they develop from the lab to like startup or industry? Like, you know, you have to redesign everything. Like, what should you be focusing on when you are in your PhD?
I think if you're in your PhD and you're looking, you know, towards this path of commercializing your own work, I think you have to get out of the lab. I think that's the first thing. You have to go talk to stakeholders, right? And that might be for us, for instance, it's talking to embryologists, but it's not just the people that are using it. It's understanding everyone in that chain, right? So the embryologists have lab managers, the doctors are referring patients for particular treatments, modalities of treatment in IVF. So they have decision-making power on what gets put in. So everyone has a different perspective on your technology. So I think the first thing is get out of the lab and you can validate the needs of your potential, future users or customers very quickly, just through a user-centered design process. I mean, everything we're doing is got a human design element to it. So the ergonomics matter, but understanding the workflow or the context that your technology is going to be in is super important, right? For example, if I made a sperm selection technology that gave me the golden sperm, the best sperm you've ever seen in your life, but it takes 12 hours, it's completely useless, right? Because you have minutes to fertilize an egg once an oocyte comes out of the body. You have maybe a couple of hours for sperm selection prep in the context of IVF, right? So you have to really get out of the lab, go understand the context, the environment that you might be playing in, and get feedback. Go talk to not just the academics and the end users and such, but go talk to other entrepreneurs, go talk to people who've exited companies. I'm not that far yet, but maybe one day. But talk to people who've done it before and get their advice.
Yeah, sounds great. That's excellent advice. Thank you.
Okay, awesome. So I don't see any more questions in the chat. So I think that's everyone's questions. Thank you both so much to Steve and Hiro for giving us two really exciting seminars today. Yeah, I hope we'll see you back at microTAStic in the future. And everyone, remember, we have more coming next month, so stay tuned for our updates. Thanks, everyone.
Thank you very much.
Thank you so much.
Thank you so much.